have our guest lecturer, Professor Yu Chen or Prof Chen, that will deliver the information and all knowledge about animal digestive system or gastrointestinal system of the animal. So maybe it will be a very valuable time for us to have Prof Chan with us today. So I hope students could pay attention to the class, and thank you very much for all lecturers who are willing to come to our session today. Maybe we can share also information and ideas on human biology, especially on gastrointestinal system. So the time will be up to 11:50 Prof Chen, or around 12:50 Taiwan time, 12:50. Okay. Okay, so if you would like to have a discussion session during the course, it will be very open for Prof. Chen.
Okay.
Okay. So the time will be yours, Prof. Chen, for the next until 12:50. Okay.
Thank you. Yeah. So thank you, Professor Yu. Kind of introduction. Yeah, I am Chen. I'm a professor in the Graduate Institute of Medical Sciences, and also I am a former director of the International Master and PhD program in medicine. So there are many students from the Indonesia to our program for the PhD and for the master degrees. So I think I'm very happy to have the opportunity to introduce the course, to have a teaching course in the Indonesia. So today, the topic I want to share with all the students and the professors is animal digestive system. The original topic is GI system, but I make a little bit modified because the biology in Taiwan is for the first year students. So we usually call it the general biology. But after discussing with the Professor Yu, I found that in Indonesia, maybe the third year student to choose the biology, so in the human biology. So I'm not sure I prepare the slides is okay or not, but I will do my best to have a clearly introduction of the digestive system in the animals. Okay, thank you. So I have two slides to very brief introduce myself. Yeah, so because it's the first time we meet each other on air, so I just want to have a brief introduction. The CV. So I'm a professor in the TMU. I also have a joint appointment professor in several programs, and my research. I also hope we can have the cooperation in the future. Maybe my research I have the four major topic in my hands. The first one is in vivo and the in vitro anti-tumor study of the natural products. As we know, Taiwan have many plants. Also, Taiwan people like to intake the Chinese herbs. So I did a lot of work about using the natural product to study the biological function of the natural products, such as the apoptosis or anti-carcinogenesis. And I also did some work in the in vivo and the in vitro anti-inflammatory studies, and also molecular anti-tumor mechanism study. And our latest publication related to lung fibrosis studies. So this is my outline of my research. Okay. So I come back to the topic. So today I want to introduce the GI system, the digestive system. So the first 5 slides I remember, I will introduce the animal body, because the animal body, the structure of the animal body needed to fit the function. So in the animal, the structure needed to fit the function. So there are 2 terms I show here. The first one is anatomy. Anatomy is the study of structure of an organism. So anatomy is like just study the structure, and the physiology is the study of the function of the organism performs. So So these two terms usually the student maybe misunderstanding the difference between the anatomy and the physiology. Anatomy is for the structure and the physiology is for the function. And also the animal structure has a hierarchy from the basic to the higher level. So the basic level is cellular level, like the muscle cell, and the tissue level, muscle tissue, and the organ level. So the cellular, tissue, organ system, and finally different systems will compose together for an organism. So the cellular level, tissue level, organ level, and the several organ will be come out to the organ system level, like the circulating system, and the organism level. Many organ systems function together to make the organism can live and can do anything. And the epithelial tissue cover the whole body, outside and inside the body. So epithelial tissue cover and lines the body and its parts. So there are several different epithelium. Like the first one is simple squamous epithelium. The simple squamous epithelium locates in the air sac of the lung. So it's very thin and easy to make the material easy to move in and move out. And the second one is simple cuboidal epithelium, forming like the tube in the kidney. And today I will introduce the GI system. So in the intensity, the epithelium is a simple columnar epithelium lying in the intensity. Okay, so in the intensity, the epithelial tissue belonging to the simple columnar epithelium. And also the stratified squamous epithelium. Stratified means the multi-layer squamous epithelium locates in the region. They are easy to the cell easy to die or to death, and they needed to proliferate to replace the upper layer. So the stratified squamous epithelium locates in the esophagus. So the GI system in the intensity, the epithelium is a simple columnar epithelium. And the connective tissue also is very important in the body. So the connective tissue binds and supports other tissues. So connective tissue can be in the biology. The connective tissue can be divided into loose connective tissue located under the skin, and the adipose tissue. Adipose tissue is for the fat storage. So store fat enlarged and the package in the cell. And the blood also belonging to the connective tissue is a fluid connective tissues. And also there are dense matrix like the fibrous connective tissue forming the ligament, and the cartilage is a strong but flexible skeletal material. And the bone also is a connective tissue, is a rigid connective tissue embedded in the calcium. Okay, so this is a connective tissue. And the muscle also muscle tissue function in the movement, and also very important parts in the GI systems. So in the GI, the muscle tissue include the skeletal muscle, cardiac muscle. But today I want to introduce the smooth muscle. So smooth muscle is in the intensity in the inside organ. So the smooth muscle lack the striations in the wall of the digestive systems, and the smooth muscle contraction is slower, much slower than the skeletal muscle. But the smooth muscle contraction, the tense is slower. It's not as strong as the skeletal muscle, but the smooth muscle contraction can sustain for a long time. So you can see the intensity or small intensity, the contraction you can sustain for a long time, but not as the skeletal muscle contraction so strong. And then I want to introduce the digestion systems. So the animal ingests their foods in the variety of ways. So all the animal eat other organisms, even dead or alive, or in peace. So in general, animal fit into one of the three dietary category. So it depends on the animal. They may be some animal only eat the vegetable, eat the grass, and some animal eat the meat, and some animal eat both. So we can divide the animal into the herbivores, carnivores, and the omnivores. So the herbivores is an animal it like to eat the autotroph. Autotroph means the plants or areas. They can produce the nutrient by themselves. So like the gorilla, cows, and the hares, and the many snails, they can eat the autotroph, so-called the herbivores. They only eat the autotroph. So these animals are herbivores. And the other, some animal only eat the other animal, like called the carnivores. Carnivores like the shark, hawk, spider, and the snake eat the other animals. And some animal eat the plants, also eat the meats, called the animals. Animals like the cockroach, bear, raccoon, and the human also, they can eat the plant and the animal, consume the plant and the animal. So we can divide the animal by their foods to the herbivores, carnivores, and the omnivores. And the animal that ingest the food in a variety of the ways. So it depends on, this is divided the animal by the foods, and this is divided by the ways to get the foods. So like some aquatic animal, like the crane is living in the water, so called the suspension feeder, because they will intake the food particle from the water. So this type of the animal called the suspension feeder. And the second one is the deposit feeder. Some animal living in the dirt or in the sediments. So they will decay the organic material consumed along the soil and the sediment. So like the earthworm. Earthworm can living in the soil, and also they eat the earthworm intake the dirt and the sediment, and the digestive the organic material for themselves. So the earthworm is a deposit feeder. And some animal living on the food, like the worm or mango living on the foods. So this type of the animal called the substrate feeder. Substrate feeder living in or on their food source, eating their way through the food, so called the substrate feeder. And some animal maybe suck the fluid from the human or from the other plants, called the fluid feeders, like the aphids and the mosquito. Both animals, mosquito suck the blood from the animal for their nutrient. So this animal called the fluid feeder. And most animal can use the teeth to spread the food to small pieces, so called the buck feeders. So most animals are buck feeders. They can eat relative large piece of the foods. So these are buck feeders. So divided the animal based on the food and based on the type of they get the nutrients. We can divide it to the animal to different feeders or to different herbivores, different type of the animals. And how the animal to digest the foods. So the food processing occur in 4 stages. So an animal want to get the nutrient to intake the foods and to support the body to live. So the animal needed to injection. Injection means the eat. The animal needed to eat the foods. So ingestion is eating, is the first stage of the digestion. And the second stage is the digestion. The purpose of the digestion is to make the large particle, large food to small foods, to small pieces or small molecule for the absorption. So the second step is digestion. So the digestion is breaking down of the food into molecules smaller enough for the body to absorb. So all the organisms using the same monomer. Why the foods needed to be digested before absorption? Because all the organisms using the same monomer, like the human and the bear and the other animal have the same amino acid and have the same fatty acid, but the macromolecules are different. So all the foods needed to digest to the monomer and to synthesize the different large molecule. So all the organisms use the same monomer, such as the amino acid. And the digestion, there are two phases. One is the mechanical breakdown. Mechanical breakdown means like a human intake the foods, needed to use the tools, use the teeth to digest, to make the large foods to pieces. And the second step, we call it the chemical breakdown. Chemical breakdown, also called the hydrolysis, needed to use the different enzyme to digest the small pieces to the monomer, and then they can be absorption. So the third step is absorption. So the cell line the digestive tract to take up the nutrient molecule and absorb the nutrient into the body. And finally, called the elimination. Elimination is undigested material passed out the digestive tract, called the elimination, like the faces or stool. Okay, so this is the four stage in the food processing, including the ingestion, digestion, absorption, and the elimination. So there is a figure in the biology book. So the foods needed to through the mouth, and the mechanical breakdown, the foods that will be mechanical broken to the small pieces of the foods. And then the chemical breakdown using the enzymatic hydrolysis to small molecules. And finally, the small molecules will be absorbed. So absorption is the third step. And the undigested material will be eliminated from the body, called the elimination. So there are 4 steps for the food processing. So the many animals using the lower grade animals using the simple body plants, like the flatworm, they only have the sac, or only have one opening. We call it the mouth. So the lower grade animal using the gastrovascular cavity as the digestion cavity. So like the hydra captures its prey, and the prey will enter the gastrovascular cavity, and the hydra intake the prey, and the prey then be partially digested by the enzyme and secreted by the gastrodermal cells. And then the small pieces of the foods will be endocytosis intracellularly. So the lower grade animal, they use because they are very simple. So they are only one layer of the cells. So they use the intracellular digestion to hydrolyze the macromolecule to the monomolecule and for their nutrients. And the undigested material eliminated through the mouth, because the lower grade animal, like the hydra, they only have the one opening called the mouth. So the food intake from the mouth, and the undigested material outside removed from the mouth. Okay, so the hydra using the intracellular digestion to produce the nutrient for their body, for them. So this is the hydra. The structure of the hydra, including the tentacle, and this is the mouth, and there is the prey. So the prey will intake from the mouth, and then the wall of the hydra only one cell. So the cell will release the enzyme to digest the prey into the small molecule, and the small food particle will engulf into the cell, and then the intracellular digestion will use the hydrolytic enzyme and produce the small molecule and for the cells. So this is the lower grade animal. They can, like the hydra, they're using the intracellular digestion. And flatworm is a little bit higher than the hydra. So they have the cyntharian and the flatworm. Most animals already have the complete digestive tract for the or alimentary canal. So the digestive tract also completed the digestive tract, also called the alimentary canal, from the mouth to the anus. So the hydra only have one opening, is mouth, and the higher animal has two open. One is a mouth, the other is a anus. So they have two openings. So the food injected through the mouth and the pharynx, okay, and through the esophagus. Esophagus is from the mouth to the stomach. So this is the pathway. So the food injected through the mouth and the pharynx and pass through the esophagus and lead to the stomach in human. And in the other animal, they have the cross gizzard for storage or stomach, depending on the species. So different species, like the birds or like the earthworm, they have the gizzard. Like the grasshopper, they have the cross. So the cross and the gizzard and the stomach are maybe the function are very similar. So they just storage the foods and soften the foods and make the foods easy to be digest. So the food injected through the mouth and the pharynx and the passages through the esophagus lead to move to the cross, gizzard, and the stomach, depending on the different species. And the cross and the stomach usually serve as the food storage organ. So that the function is for the food storage. And the gizzard in some animals, they have some stone in the gizzard. And the function of the gizzard is to grind and the fragment of the foods. And then after the storage and after the grind the foods, and then the foods will move to the intensity. In the intensity is very important for the digestion. So the intensity, the digestive, there are several digestive enzymes in the intensity. The enzyme will catalyze the hydrolysis of the foods to small monomer, and the undigested waste are eliminated through the anus. So the system enables the organism to ingest additional foods earlier meal completed the digest. So we have the long digestion organ. So this system, when the foods from the mouth, they will move into the stomach, move into the intensity, but the foods are not well completed digestion. We can intake the more foods. So the digestive system can enable the animals to ingest additional foods earlier the foods are completed digested. So this is the important function of the digestive systems. So this slide will show the three different animals. This is the earthworm. We can just very quickly to see the digestion systems from the mouth, pharynx, esophagus, crops and gizzard. This for the storage of the food. This for the grind the food. And then the food will move into the small intensity. The intensity can release the digestion enzyme to catalyze the hydrolysis of the foods. And then the anus. So there are two openings, one food intake from the mouth, and the undigested material will remove from the anus. And this is the grasshopper, also the same. They have the mouth and the esophagus and the cross gizzard. And in the grasshopper, there is a gland called the Gatry pouch. Gatry pouch can release the enzyme and the stomach and the intensity and the anus. So they also have the two openings. And the birds also the same, from the mouth, esophagus, stomach, crop, gizzard, and the intensity and the anus. So most animals has a complete digestive systems from the mouth to the anus. Okay, so this called the alimentary canal. Alimentary canal. So the digestive tract also called the alimentary canal. So the following, I want to introduce the mammalia. Mammalia, like the human, is the highest level of the animals. So the mammalia digestive systems consist of the alimentary canal and very accessory glands, and secreted the digestive enzyme into the canal through the ducts. So there are several terms. Usually we can say the peristalsis. Peristalsis is the movement of the digestive systems. So peristalsis is a rhythmic wave of the contraction of the smooth muscle in the wall of the canal. And like the esophagus or intensity, they will peristalsis. They will move the foods forwards. So the peristalsis is often to be found by the smooth muscle in the wall of the digestive systems. They will push the food along. And in the digestive systems, there are some muscle can regulate the foods. So for example, in the food from the esophagus to the stomach, they are sphincter. Sphincter is a muscle, muscular ring-like wave. They will control the foods. And when the food is ready, they will open in the sphincter and make the food move down. So the sphincter is a muscular ring-like wave, regulate the passage material between the specialized chamber, such as like from the esophagus to the stomach, from stomach to the small intestine. So this control the food to move down is by the sphincters. And in the digestion systems, there are several accessory glands, including the salivary gland. Salivary gland in the mouth. Salivary gland will release the amylase to digest the carbohydrates. And the pancreas is very important digestive organs, and the liver and the gallbladder. I will introduce the function of this gland later. So in the digestion, the first action is the chewing and the swallowing. So when we intake the foods in the mouth, we needed to take around, it takes around 5 to 10 seconds to chewing and the swallowing, and the foods will move from the mouth to the esophagus. So the chewing and the swallowing, it takes to about 5 to 10 seconds, and the food to pass down the esophagus to the stomach. And then in the stomach, the food will stay in the stomach about 2 to 6 hours, being the partially digest. So in the mouth, the food will take around 5 to 10 minutes to chewing and the swallowing, and in the stomach, the foods will stay in the stomach around 2 to 6 hours. And finally, digestion will occur in the small intestine. So the small intestine is an important organ for the digestion. So in the small intestine, the foods will take around 5 to 6 hours. And about 12 to 24 hours, the undigested material will pass through the large intestine, and the feces are expelled through the anus. Okay, so this is the process and the time. It's not so accurate, but it takes around the time 2 to 6 in the stomach, 5 to 6 in the small intestine, and 12 to 26 hours in the undigested material will be expelled through the anus. So this is the organ, the glands or the organ of the digestion in the human. So in the mouth, there is a important salivary glands. So in the mouth, there is the tongue. Tongue is very important because tongue can mix the food well in the mouth. And the parotid gland, and the sublingual gland, and the submandibular gland. So this gland compose the salivary gland, and release the enzyme, and release some some mucin. Mucin is a liquid. Is a liquid that can protect the mouth. Okay, so the salivary gland, and this is oral cavity, and from the food from mouth through the pharynx, and then this is the esophagus, and the move from the esophagus through the peristalsis, move to the stomach. So the stomach has a two junction. One is connected with the esophagus. One is connected with the small intestine, duodenum. So there are two sphincters here. The connection between the stomach and the esophagus called the cardiac orifice. And the connection between the stomach and the duodenum called the pyloric sphincter. Okay, so from the stomach, they will connect it, move to the small intestine. Small intestine, there are three parts, including the first part is the duodenum. Okay, so the duodenum, ileum, and the ileum. So the small intestine, and then to the large intestine, and finally to the anus. So large intestine, the end of the large intestine linked with the anus is the rectum. Okay. And the small intestine and the large intestine, there is the junction here. This junction also the second. And the end of the second layer is like a finger-like structure called the appendix. Okay. So this is the digestive systems in the humans. Also there is the liver. Liver will release some enzyme, some buyer to support it to digest the fatty acid. And the gallbladder is the organ to collect the bile. And the pancreas locates behind the stomach. Pancreas will release the several hydrolytic enzyme to the small intestine and to digest the many different molecules. Okay, so this is the structure and the organ of the digestion in human. So we can see the wording here. So the digestion begin in the oral cavity. So both physical and the chemical digestion of the food begin in the mouth. So in the mouth, the food in the mouth will be chewing, and the teeth of the berry shape cut smash the foods to the small pieces, and then easier to swallowing. So the mouth also is very important for the digestion, because we intake the foods, and the foods maybe they are several sharp angles and not easy to swallowing. So in the chewing and the tongue, we will mix the food well, and we also have the saliva. Saliva can mix with the foods and make the foods easy to be swallowed. So the presence of the food in the oral cavity triggered by nervous reflex and cause the salivary gland to deliver the saliva to the mouth. Okay, so the mouth is the beginning of the digestion. So we can see the structure of the mouth. So this is the incisor, the teeth, the incisor, this is canine, this is molar, this is wisdom tooth, and this is the tongue. So there are two salivary glands here, and they're opening, saliva opening. So when the foods in the mouth, they will stimulate, they will induce the saliva release to the mouth, and then the foods will be partially digest, and the tooth will grind the foods and fragment the foods to more small pieces, and the mix well with the saliva and easy to be swallowed. Okay, so the opening of the salivary gland here, and this salivary gland, and this tongue, and this tooth, different teeth here. And we can see the different type of the animal, the structure of the tooth are different. So you can see here. So this is the carnivores. Carnivores like the dog and the cat families. They have the large canine, and the large pointed incisor and the canine, and the jagged premolar. This is jagged like a premolar and the molar structure here. So the carnivores have this type of the teeth in the mouth. And the herbivores always eat the grass or eat the small trees. So they have the very well developed molar and the premolar. So the premolar and the molar reach the surface to grind the plants. Incisor and the canine modified for biting the piece of the. So the incisor just modified for biting the piece of the plants. And some of the herbivores like the canine here. So some of the herbivores the canine or did not find. And the human have 32 teeth and the 4 blade-like incisor and a pair of the canine and the 4 premolar and the 6 molar. Okay, so this is for the omnivores. Human are omnivores. So this I just show the different type of the animal depends on what kind of the food they intake. Like the carnivores, herbivores, the structure of the teeth are different. So the saliva is very important for the digestion. Saliva is a beginning and occur in the mouth. So the saliva contains a slippery glycoprotein called the mucin. So the mucin can protect the mouth because sometimes we eat the foods and the foods are very sharp, so easy to make the inside the mouth easy to make it hurt. So saliva can protect the soft lining of the mouth from the abrasive and lubricate the foods for easy to swallowing. And the saliva also contain the buffer. Buffer can prevent the tooth decays by neutralize the acid in the mouth. And also in the saliva also contain the antibacterial agents. So when we eat some foods, maybe the food has some bacteria or is not so clean. So the saliva also can contain the antibacterial agent. They can kill the bacteria and make the foods easy, clean, and safe to intake. Okay, so the saliva has a function like the slippery glycoprotein, easy to swallowing, and the buffer to protect the tooth, and also has the antibacterial agents. And in the mouth, the carbohydrate will be primary digestion. So chemical digestion of the carbohydrate is the main source of the energy beginning in the oral cavity. So the saliva contains the amylase, salivary amylase. This is an enzyme for hydrolysis of the starch and the glycogen into the small polysaccharide or disaccharide, maltose. So in the carbohydrate will be initially digested by the amylase in the mouth. So the starch and the glycogen will be digested into the small molecular, smaller, but not the monomer. It's smaller polysaccharide and the disaccharide, like the maltose. And the tongue in the mouth also be very important. So the tongue will taste the foods. They taste the foods, and in the chewing, the tongue will mix the foods and make the foods into a bowl. We call it the bolus. Okay. So the tongue will taste the food and manipulate during the chewing and shape the food into the bowl called the bolus. And the bolus is easy to swallow. So the tongue will push the bolus back in the oral cavity and into the pharynx. Okay. And the pharynx is an important junction because in the pharynx there are 2 openings. One is for the esophagus, the other is for the trachea. Trachea is a wide pipe. So in the pharynx there are 2 openings. So one is for the esophagus, the other is for the trachea. It's for the air. Okay. So in the pharynx there is an opening. We call it the glottis, and the blockage by a cartilaginous is a cartilage-like. It's called the epiglottis. So the glottis and epiglottis will control the opening of the trachea and control the food safely move into the esophagus, not into the trachea. If the food into the trachea, they will cause the very serious, uncomfortable, and maybe it will induce the inflammation of the lung. So the mechanism normally ensure the bolus will guide into the entrance of the esophagus and not direct down into the wide pipe. So the epiglottis is very critical in the swallowing. Some patients, some people has a problem in the normal function of the epiglottis. So they eat the foods. If the foods will move into the wide pipe, trachea will cause the serious problem. So how the epiglottis to control the food swallowing. So when the people not swallowing, the esophageal sphincter muscle contracts and the epiglottis is up. So the glottis is open. So the airway is open. So no swallowing, no food intake. The esophagus sphincter muscle contract and the epiglottis up and the epiglottis open and the airway is closed. And when the food bolus reaches the pharynx, the larynx will be up and the epiglottis will cover the glottis and close the trachea. And the esophageal sphincter will release and the bolus will enter the esophagus. Okay, so then because the trachea has been covered by the epiglottis, so the food will not enter the trachea. The food will enter the esophagus. So in the meantime, the larynx move down and the trachea will reopen and the food will move to the stomach using the peristalsis. Peristalsis I introduced before, like the smooth movement from the upper to the lower of the esophagus. So there is the figure here. So in the swallowing, if no foods in the mouth is not swallowing, the esophagus, there is a sphincter here. So in the esophagia, there is a trachea opening and also is a esophagus. So there is a glottis here. So in the esophagus, there is an important structure called the smooth. There is a sphincter control the food enter the esophagus. So it's not swallowing. The esophageal sphincter contracted. So the esophagus is closed and the trachea is open. So esophageal sphincter muscle contracted and the glottis is open. So the air can move into the lung by the trachea. So no swallowing, the airway is open. And when the food move to the pharynx, the larynx will move upward, will cover the trachea. So the epiglottis cover the glottis and prevent the food into the trachea. And then the esophageal sphincter will be released, will be opened, and the food will slide into the esophagus. Okay, so if the food reach the pharynx, the larynx move upwards, epiglottis cover the glottis, and prevent the food into the trachea. Esophageal sphincter release, and the food will move into the esophagus. And then the food from upper of the esophagus to the lower of the esophagus reaches the stomach using the peristalsis. Okay, there is a slide to show how the muscle control the peristalsis here. So the food enter the esophagus and will move down to the stomach. So the esophagus conduct the food from the pharynx to the stomach by the peristalsis. So this term is very important in the biology, like the smooth movement of the food. So how the peristalsis occur in the esophagus? So there are two muscles control the peristalsis. So the muscle at the top of the esophagus are striated, and under there are two different muscles. I will show you the slide here. So in the esophagus, there are two different types of the muscle. In the inside is a circular muscle layer, and the outside is a longitudinal muscle layers. So there are two types of the muscle in the esophagus. So if the bolus in the esophagus, we can see there are two different muscles here. The outside is longitudinal, the inside is a circular muscle. So in the upper parts of the bolus, the circular muscle will be contracted. So the circular muscle contracted, and will squeeze the food move down. And in front of the bolus, the longitudinal muscle will be contracted, will shorten the distance between the bolus and the stomach. Okay, so repeatedly, the upper parts, circular muscle contracted, squeeze the food move down, and the longitudinal muscle before the bolus, these parts reduce the distance between the bolus and the stomach, and finally the food will be moved into the stomach. So the relaxed muscle, circular muscle, contracted passageway push the bolus down, and the longitudinal muscle contract, shortening the passage, and then finally the food will reach the stomach. Okay, so this is called the peristalsis in the stomach, using these two types of the muscle. One is the circular muscle, the other is longitudinal muscle layers. And then the following, I will introduce what happened in the stomach. So the stomach, the function of the stomach is to store the foods and perform the preliminary digestion. So the stomach locates in the upper abdominal cavity, just upper the diaphragm. Okay, diaphragm. So the stomach can store the 2 liters of the foods and the fluid, and store the entire meals. So the stomach also can secrete the digestive fluid, called the gastric juice. Gastric juice means like including the HCl and including the pepsin, is an enzyme by the stomach. So the stomach can secrete the digestive fluids and mix the secretion with the foods, and then they will digest the protein, preliminary digestion of the protein in the stomach. So the gastric juice secreted by the epithelium lying in the deep pits in the stomach walls. So in the stomach, there is a high concentration of the hydrochloride. Hydrochloride is a HCl. So the pH value of the stomach gastric juice is about 2. So this is very acid. It's acidic enough to digest iron nails. So this acid disrupts the extracellular matrix and bind the cell together. So in the stomach, the pH value is very low, it's around 2, and the condition is acidic condition. So this condition can kill the bacteria that are swallowing with the foods. And in the stomach, there is in the gastric juice, there is an important enzyme called the pepsin. The pepsin is a protease. The pepsin can recognize two amino acids, is a phenylalanine and the tyrosine. So the pepsin can digest at the site of the phenylalanine and the tyrosine. So this enzyme can hydrolysis of the protein. So the pepsin can work well in the acid environments, and the black peptide bond adjacent to the specific amino acid. Specific amino acid is phenylalanine and the tyrosine, and the digested the protein to small polypeptide, not amino acid. Okay, so the pepsin in the gastric juice can digest the protein very close to the phenylalanine and the tyrosine, and produce the small polypeptide. So the pepsin secreted by the by the wall of the stomach. But initially, the wall secreted the pepsin is in the inactive form, called the pepsinogen. So you can see if a gen at the end of the words means the inactive is an inactive form. So the stomach wall release the pepsinogen, and then there is a chief cell in the stomach wall will release the HC, the enzyme to activate the pepsinogen to pepsin. So we can see the figure here. So this is the stomach wall. This is the stomach wall here. So they are pit here. So there are two different cells. The red one is mucus cells, and the orange one is chief cells, and the yellow one is parietal cells. So in the deep of the pit in the stomach, there is the chief cell will release the pepsinogen. Pepsinogen is an inactive form of the pepsin. So and the parietal cell will release the HCl. So the pepsinogen will be digested, will be cleavage by the HCl and make the inactive to the active. So the pepsinogen will be, it is an inactive form, will be digested by the HCl, cleavage by the HCl to produce the pepsin. So the pepsin is an active enzyme, active protease. And the pepsin also can digest more pepsinogen to produce pepsin. So the step 3, we call it the positive feedback. Positive feedback. So initially, the pepsinogen will be activated by the HCl. And finally, to produce the pepsin. And the pepsin can positive feedback to digest more pepsinogen to pepsin. Okay, so the parietal cell in the pit secrete the hydrochloric acid and convert the pepsinogen to active pepsin. And then the pepsin can digest more, can activate more pepsinogen molecule. So this pathway, this process in the biology is very often to see the positive feedback system. So in the biology system, we can see the positive feedback produce more, and if the inhibit, we call it the negative feedback. So there are two different feedback. One is positive, the other is negative in the biology systems. But in the stomach, the pepsin can produce more activated enzyme called the positive feedback. So the stomach can defense. So the stomach is the stomach condition is very acidic. So the stomach coating of the mucus secreted by the epithelium, and they can produce the stomach lining from the HCl digestion damage. So there is a disease called the gastric ulcer. Gastric ulcer means some damage, some hurts, some damage in the stomach lining. So the gastric ulcer in the stomach lining caused by the acid tolerance bacteria, Helicobacter pylori. So gastric ulcer caused by the HP bacteria. HP bacteria is very special bacteria. It can live in the acidic condition. So the ulcer often treated with the antibiotics. Okay, so gastric ulcer is a lesion or damage in the stomach lining. So by the acid tolerance bacteria, HP. So the ulcer often treated with the antibiotics. So the stomach will be tuning every 20 seconds. So all the nutrient will be break down by such as the cheme. So the foods move from the esophagus to the stomach. In the stomach, they will use the HCl, use the pepsin to preliminarily digest the foods, and finally the foods will convert it to the acid cheme here. So the acid cheme is ready for use in intensity. So the acid chemie will move down to the small intestine. Okay, so most of the time of the stomach is close of the either ends. We told before there are two ends. One is the stomach junction, the junction between the stomach and the esophagus. The other is the junction between the stomach and the small intestine. So the opening esophagus and to the stomach called the cardiac orifice. And the other is a pyloric sphincter. Pyloric sphincter is to control the food from the stomach to small intestine. So opening from the stomach to small intestine is the pyloric sphincter. So after the foods in the stomach be digested and converted the food to acid chemie, the pyloric sphincter will be opened, and the acid chemie will move from the stomach to small intestine. So about 2 to 6 hours after for the stomach to be empty. So then the acid chemie will move to the small intestine. So the small intestine is the major organ for the digestion and the absorption. So in the human, the length of the small intestine about 6 meters, and the small intestine is the longest section of the alimentary canal. So in the digestive canal, the small intestine is the longest part. So the most of the enzymatic hydrolysis of the food, the macromolecular, and most of the absorption of the nutrient into the blood occur in the small intestine. So this means the small intestine is is the most important part of the digestion. So because the digest the foods into the monomer and the absorption of foods occur in the small intestine. So in the small intestine, the first 25 centimeter called the duodenum. Duodenum, the acid chemie from the stomach will mix the digestive juice in the duodenum. In the duodenum. So in the duodenum, there are many digestive enzymes released from the pancreas, liver, gallbladder, and the glands of the small intestine walls. They are mixed here. So you can see here. So this is the 25 centimeter in the beginning of the small intestine called the duodenum. So the duodenum, there are the liver will release the bile into the duodenum, and the gallbladder is a collector, is an organ, to collect the bile. And as the chemie from the stomach will move into the duodenum, and the pancreatic juice. Pancreas is an important digestive organ, so release several enzymes into the duodenum. So the duodenum is the most important part in the small intestine, because in the duodenum there are many different enzymes, different molecules, like the bicarbonate. They will neutralize the acid chemie here. So the pancreas produces several hydrolytic enzymes, and the alkaline is an alkaline solution, like the bicarbonate will buffer the acidity from the chemie. So they will mix here. So this is a very important part in the digestion, is the duodenum in the small intestine. So the liver is offer the action of the liver in the digestion. So the liver perform a wide variety of the important function. So the liver will produce the bile. So the bile is stored in the gallbladder until needs. So it contains the bile salts. Bile salts are detergents. They can aid the digestion and absorb the fats. So the bile also contains the pigments, like so the pigment is a byproduct of RBC. It's a red blood cell destruction in the liver. So the old senescent RBC will be destroyed in the liver. So the pigment, the bile pigment from the RBC. So the bile pigment are eliminated from the body is the feces. So you can see the feces color is from the bile pigments. And the digestion of the carbohydrate, as I mentioned before, the carbohydrate initially digested in the mouth, the saliva. Saliva contains the amylase, right? So the digestion of the starch and the glycogen began by the salivary amylase in the oral cavity and the continuous in the small intestine. So the pancreas can release the amylase, hydrolyze the starch and the glycogen and the small polysaccharide into the disaccharide. So the pancreas also release the amylase and the hydrolyze the starch and the glycogen to the disaccharide. A family of the disaccharide also is a little bit larger and cannot be absorbed. So the disaccharide should be digested to the monosaccharide. So the disaccharide should be digested by the disaccharide. Disaccharide is an enzyme to convert the disaccharide to the monosaccharide. So the hydrolyze each disaccharide into the monosaccharide. There are two important disaccharides. One is the maltase. Maltase can split the maltose into the two glucose. Glucose is a monomer, so we can absorb the glucose. And the sucrase can split the sucrose into the glucose and the fructose. So converted in the small intestine, converted the polysaccharide to the disaccharide, and the disaccharide can be catalyzed by the maltase or sucrase to produce the glucose and the fructose. And the glucose and the fructose are monomer, so can be absorbed from the small intestine walls. So the intestine epithelium can be site for the sugar absorption, because in small intestine, all the carbohydrate can be converted to the monomer. Okay. And the protein preliminarily digested in the stomach, right? So in the stomach, the protein can be digested to the small peptide, not a monomer. But in the small intestine, the small peptide can be converted to the amino acid and it can be absorbed in the small intestine. So the several enzymes in the duodenum dismantle the polypeptide to the amino acid. So there are several different enzymes here. In the stomach, we know the enzyme is pepsin. Pepsin can digest the phenylalanine and the tyrosine very close to these two amino acids. In the small intestine, there are three proteases. The first one is trypsin and the chymotrypsin. Trypsin can recognize the lysine and the arginine. Chymotrypsin can recognize the tyrosine and the tryptophan. So they can digest the large polypeptide to small one, not a monomer, okay? And the other enzyme is dipeptidase. Dipeptidase attached to the intestine lining and spread the small chain. And finally, there are two important enzymes. One is the carboxyl peptidase, the other is amino peptidase. Carboxyl peptidase can remove the amino acid from the C-terminal. As we know, the peptide contains the two functional chain, two functional group. One is a carboxyl group, another is the amino group. So in the small intestine, the carboxyl peptidase can remove the amino acid from the C-terminal, and the amino peptidase can remove the amino acid from the N-terminal. So the carboxyl peptidase and the amino peptidase can split off one amino acid from the C-terminal and the N-terminal. And finally, the small peptide can be converted into the amino acid for the absorption. Okay, so in the small intestine, the peptide can be converted to the amino acid and for the absorption. So there are several enzymes here: trypsin and the chymotrypsin and the dipeptidase, and finally the C-terminal peptidase and the N-terminal peptidase converted the peptide from the C-terminal and N-terminal to the amino acid. So there is a figure here. So you can see the So the many of the protein digestive enzymes, such as the aminopeptides secreted by the intestinal epithelium, and the trypsin, chymotrypsin, and the carboxyl peptides are derived from the pancreas. So, and this is the different parts release the different enzymes. So the aminopeptides from the epithelium of the small intestine, and the trypsin, chymotrypsin, and the carboxyl peptides secreted in the inactive form by the pancreas. Inactive form like I mentioned in the stomach, like the pepsin. The inactive form is the pepsinogen. So the wall of the stomach wall release the pepsinogen, and then pepsinogen is inactive form, can be converted to the pepsin and the positive feedback. So this is also the same. So the pancreas release the trypsinogen and the carboxyl peptides precursor called the pro-carboxyl peptides and the chymotrypsinogen. So these three enzymes from the pancreas are inactive form, and then some induced inactive form to the active form. So there is another intestinal enzyme called the internal peptides. Internal peptides derive from the intestinal walls. So the membrane bound internal peptides, this enzyme can catalyze the inactive trypsinogen to the trypsin, and then the trypsin can positive to induce, and also can induce the pro-carboxyl peptides to carboxyl peptides in active form, and the chymotrypsinogen to the active chymotrypsinogen, and to catalyze the digestion of the protein. Okay, so the active trypsin then active the other two. Okay, so this is the process for the enzyme activation in the small intestine. So in this third slide, I want to make clear the several enzymes involved in the protein digestion. Okay, so aminopeptides from the intestine, and the trypsin, chymotrypsin, and the carboxyl peptides from the pancreas. And the initially release is inactive form, and the internal peptides can catalyze the inactive trypsinogen to the trypsin, and then trypsin can catalyze the other two to make a more active enzyme. Okay, and then the next one is the digestion of the nucleic acid. The nucleic acid, like including the DNA and the RNA, right? The OC nucleic acid. So the digestion of the nucleic acid involved in the hydrolytic assault, similar to the protein. So a team of the enzyme called the nucleus. Okay, so the nucleus is the enzyme to catalyze the digestion of the nucleic acid. So the nucleus hydrolyzes the DNA and RNA into the components of the nucleotide. As we know, the nucleotide, including the three parts, is a base, is a phosphate group, is a five pentose, is a pentose, right? So neither the nucleus needed to hydrolyze the DNA RNA into their component, and the component can be absorbed by the small intestine. So the other hydrolytic enzyme then break the nucleic acid down to further nucleoside, nitrogenous base, and the sugar and the phosphate. So like the nitrogenous base and the sugar is a pentose, and the phosphate is a small molecule, so can be absorbed. And the digestion of the fat, until now, the fat can not digest by the stomach, not digested by the small intestine. So here, the fat will be digested into the fatty acid. But the fat is not easy to be digested because fat is not water soluble, so needed to be emulsification. So make the fat easy to be dissolved in the water, and the enzyme can be used to catalyze the digestion. So near all the fat in the meal reach the small intestine undigested. So this is different. Is a different parts, different from the carbohydrate and the protein. So normally the fat molecules are insoluble in water. So this is the big problem for the fat digestion. but biosalt really secreted from the gallbladder into the duodenum. They will coat the fat droplet, and this process called the emulsification. So the large surface area of this small droplet is exposed to the lipase, and after the emulsification, the fat can be slightly maybe soluble in the water, and the lipase is an enzyme can hydrolyze the fat molecule into the glycerol, fatty acid, and the glycerol. And the glycerol, fatty acid, and glycerol can be absorbed by the small intensity. So you can see here. So the TG triglyceride are break down to the fatty acid and the monoglyceride by the lipase. So this triglyceride can be break down to the fatty acid and the monoglyceride, and then the small molecule, the monomer, can be absorbed by the small intensity. And the fatty acid and the monoglyceride can be absorbed by the small intensity, and then in the small intensity, the epithelial cell, both of them will be composed, will be get together to the triglyceride and form the chylomicron and into the lactea, and move to the part of the body. So the monoglyceride and the fatty acid diffuse into the epithelial cell and reform into the triglyceride. And the triglyceride are incorporated into the chylomicron. Chylomicron enter the lactea, are carried by the lymph to enter the lymph systems, and then can be carried out, can carry to another parts. So this is for the fat digestion. So most of the digestion occur in the duodenum, and I mentioned in the maybe the slide, there are three parts of the small intensity. The first 25 centimeter is duodenum, and the other two sections are jejunum and the ileum. So the jejunum and the ileum function mainly in the absorption of the nutrient and the water. So the first part is for the digestion, and the jejunum and the ileum function for the absorption of the nutrient and the water. So to enter the body, the nutrient in the lumen and the pass through the line of the digestive tract. The small intensity has a very huge surface area around the 300 meters square, roughly the size of the tennis court. I just mentioned if you can count the area, the small intensity has a huge surface area around the 300 meters square. So there is a table to make the collection of the enzyme. So in the carbohydrate in the digestive system, from the starch is a polysaccharide, and the pancreatic amylase to convert it to the starch to the maltose. Maltose is a disaccharide, and then the disaccharide using the maltose, sucrase, and the lactase to convert it to the disaccharide to the monosaccharide. And the monosaccharide is the monomer of the carbohydrate, and it can be absorbed. And the protein in the small intensity, the polypeptide can be digested by the trypsin and the chymotrypsin to small polypeptide. And the small polypeptide or dipeptide can be catalyzed by the aminopeptidase, carboxyl peptidase, and the dipeptidase to the amino acid. Amino acid is the monomer of the protein, so can be absorbed by the small intensity. And the nucleic acid also including the DNA and the RNA, both of them can be digested by the nucleus to the nucleotide. And the nucleotide can use in the other enzyme to catalyze the digestion of the nucleotide to the nitrogenous base, sugar, and the phosphate, and they can be absorbed by the small intensity. And the fat globular needed to be emulsification by the biosalt to increase the solubility, and to fat droplet emulsified. And the fat droplet can be digested by the lipase, and to release the fatty acid and the glycerol, and to form the chylomicron, and to use in the lacteal lymph system to move to the another parts. So this is enzyme digestion in the small intensity. And then I want to introduce the structure of the small intensity. So the anomalous surface of the small intensity is an adaptation greatly increase in the rate of the neutron associates. So the small intensity, as shown in the previous slide, the total area around the 300 meters square. So because the small intensity, the major function is to absorb the nutrient into the body. So the large circular force, so they are in the surface of small intensity, there are many finger-like projections called the VD. And each VD, using the microscope, you can see that there are many small VD called the micro VD in the VD. So the large circular force in the finger-like projection called the VD, and each virus has many micro VD on the surface.
No.
Ada?
Ada.
Ya enggak, cuma ngecek aja, saya coba download.
Is okay?
No, salah. Download saya salah.
So can I keep going?
Yes, Prof, please.
Okay, yeah. If you have any questions, you can tell me, or the student or the professor can let me know. Okay. So the penetrating of the core of the virus is a net. In the core of the virus, there is a net of the microscopic blood vessel, capillary, and the single vessel is a lymphatic system called the lacteal. Okay. We can see the structure here. So this is the structure of the small intensity. So in the world of the small intensity, there are many VD here. So this is intensity world, okay. And in the virus, we can see the many micro VD. So in the VD, there the capillary network here, and in the central part, the yellow one is the lacteal systems. So the nutrient, the monomer, can be absorbed from the outside of it enter into the capillary network, and then the fatty acid can move into the lacteal systems, and then carry the material, carry the nutrient to the whole body. Okay. So how the material move into the capillary, how the material move into the lacteal systems. So there are different ways, including the passive transportation. So the passive transportation depends on the concentration. So through the concentration gradient, the material can move from the higher concentration to the lower concentration parts, so called the passive transportation. So such as the fructose. So the transport of the nutrient across the epithelial cell down layer concentration gradients from the lumen to the small intensity epithelial cells into the capillary. So this is called the passive transportation. So the material using the passive transportation by the concentration gradient, they don't need the ATP, don't need the energy. But some material are very precious. So although the concentration is not higher, but we needed to reclaim into the body. So another is called the active transportation, like the amino acid, small peptides, vitamin, and the glucose. These material are very important for our body. So our body cannot exclude them. So these material are pumped against the concentration gradient by the epithelial membrane, called the active transportation. So the active transportation allows the intensity to absorb a much higher proportion of the nutrient in the intensity. Then would there be possible passive diffusion? Okay. So the active transportation needed to use the ATP, use the energy, and they can against the concentration gradients. So the glycerol and the fatty acid can be absorbed by the epithelium and recombine into the fats. So the fats are mixed with the cholesterol and coated with the special protein to form a small globular called the chylomicron. So the capillary and the vein drain the nutrient away from the villi, converge into the hepatic portal vessel. So the material enter into the capillary and then converge into the hepatic portal vessel, and then to the liver. Okay, and the fatty acid with from the chylomicron enter the lacteal systems. And then the next part of the digestion is the large intensity. So the reclaim of the water is the major function of the large intensity. So the large intensity also called the colon. Colon is a part to reclaim the water. So the junction between the large intensity and the small intensity, the junction between these two parts is a T-shaped junction, because the shape like the T. So there is a sphincter control the movement of the material. So the T shape is called the second. So there is a second is a junction between the small intensity and the large intensity. So there is a finger-like extension called the appendix. The function of the appendix is not so clear, but there is well known the appendix. There are many macrophage or the immune cell will stay in the appendix. So they predict the appendix maybe make a minor contribution to the body defense. Okay, so the appendix is an organ, but the function is not so important. But there are paper reported that the appendix, there are some immune cell or storage in the appendix, so make a minor contribution to the body defense. So the major function of the colon, the large intensity, is to recover the water. So maybe a person every day, each day, will intake 7 liters of the liquid into the lumen of the digestive tract, and about the 90% of the water is reabsorbed, and the most water absorbed in the small intensity, and the rest in the colon. So the 90% of water will be reabsorbed, and the most water reabsorbed in the small intensity, and the rest in the colon. So the digestive waste, the faces become the more solid and move along the colon by the peristalsis. So as mentioned before, about the 12 to 24 hours, the material, undigested material, will remove from the body. So the diarrhea is a term for the digestive problem, resulted in the insufficient water is absorbed, and the constipation is too much water is absorbed. The faces is too solid, cannot be easy to expel to the body. So the diarrhea is insufficient water absorbed. Constipation is too much water is absorbed. And in the large intensity, there is another function. There are many bacteria, many flora of the most bacteria, harmless bacteria will live in the large intensity. So living in the large intensity, there is a rich flora of the harmless bacteria living in the large intensity, and they will produce some minor material like the vitamin, or can support the body. So one of the most common inhabitants of the human colon is E. coli. E. coli is a favorable research organism. So the E. coli is a very common material for the research in the lab, because all the genome of the E. coli is well known. So we can make the genetic manipulation in the E. coli using the plasmid or some manipulation to produce the recombinant E. coli to produce the material for human means or for your research. So the byproducts of their metabolism, many colon material generate a gas, including methane and the H2S hydrogen sulfate. So the bacteria in the colon will generate generate the gases, including the methane. Methane is CH3, CH4, and the hydrogen sulfide is H2S. And the bacteria also produce the vitamin, including the biotin, folic acid, vitamin K, and the several B vitamin. Supplement is very critical supplement for our daily intake of the vitamin. So the bacteria, harmless bacteria living in the large intensity can provide the vitamin for our needs. So this is the T junction, okay? This is small intensity, the large intensity, and the junction between the junction here, like the T shape, and called the second in the large intensity, and at the end, this is a small finger-like a structure called the appendix, okay? And the phases is the products of the digestion. Undigested material will be removed from the body. So the phases contain the masses of the bacteria, and the undigested material, including the cellulose, will be expelled, will be removed from the body. The cellulose fiber has no calorie value to the human, so the human cannot use the cellulose because we did not have the cellulose to digest the cellulose. So the phases maybe also contain the excess salt and are excreted into the colon. And the terminal portion, the large intensity, the terminal portion of the large intensity is the recon. Recon is the storage for the phases. So the phases are stored in the recon. And between the recon and the anus, there are two sphincters to control when the phases can be moved out. So the recon and the anus, they are two sphincters. One is involuntary, the other is voluntary. So in the control, there are two sphincters, involuntary and voluntary, can control when the anus will be open, will close, and to control the phases move out. And different animal has a different length of the digestive systems. So the length of the vertebrate digestive systems is also correlated with the diet. So as I mentioned before, the herbivores and the carnivores using the different food sources. One is for the one is use the plant and the energy. The other is for the meat. So the herbivores and the animals has all longer elementary canal. So you can see the two animal is a carnivores. Carnivores is for meat, is the food from the meat, and the herbivores. These herbivores, koala, the plant is the food. So you can see there the koala has a longer digestive system, digestive tract. So the herbivores has a longer elementary canal, because the vegetation is more difficult to digest than the meat because it contains the cell walls. Because the herbivores intake the plant, and the plant cell has a cell wall, so it needed to take a long time. It needed some bacteria to help them to digest the cellulose. So the vegetation is more difficult to digest. So the herbivores has longer elementary canal than the carnivores. And in the vertebrate, there are symbiotic microorganisms can help nourish the vertebrate. So much of the chemical energy in the digestion of herbivorous animals is contained in the cellulose of the plant and the woods. So animals do not produce the enzyme to digest the cellulose. So many vertebrates solve the problem by housing the large population of the symbiotic bacteria and the protists in the elementary canal. So these two sentences I just want to mention. The animal intake the plant, intake the cellulose, but although intake a large amount of the cellulose, but there is no enzyme can digest. So some animals, some herbivores, they will house the large population of the symbiotic bacteria or protists in the alimentary canal. So this microorganism have an enzyme, can produce the enzyme to digest the cellulose to small simple sugar and for the animal, and it can be absorbed. So the symbiotic bacteria of the rabbits and some rodent living in the large intensity and the second. So the herbivores has longer alimentary canal, has longer second and the longer large intensity because they needed to the space for the bacteria growth inside. So the most the nutrient absorbed in the small intensity, and this organism recover the nutrient from the fermentation. Fermentation in the large intensity, they can fermentation and make the cellulose easy to be digested. So the koala, as I mentioned here, koala, koala has a longer second, here. So the koala has an enlarged second, and the symbiotic bacteria ferment finally shredded the leaves. So the herbivores intake the plants, so they needed to have well digested the plants and digest the cellulose. So they evolution create a special way for the herbivores, called the ruminants. So ruminants include the deer and the cattle and the sheep. So when the cow first chews and swallowing the grass, so the rumen, like the cow, the cows has a full stomach. One is the rumen, the other is the reticulum, omasum, and the abomasum. So you can see the rumen, the cow has a full stomach. So after the cow intake the grass, right? The food will enter the rumen first. You can see the green line here. The food will enter the rumen and then enter the reticulum and then go back to the mouth and the rechewing. After the rechewing, the foods will go back to the omasum and then to the abomasum and then to the small intensity, large intensity to the anus to expel. So this animal called the ruminant. So the cow swallows the cud and move into the omasum where the water is removed, and then into the abomasum. Abomasum, the function of the abomasum is to digest by the cow's enzyme, and then the cellulose can be moved to the small intensity for the digestion and the absorption. So this animal has a full stomach, and the evolution to create this special way is for the cellulose digestion, and to make the animal can get enough energy, get enough nutrient they needs. And then I want to mention about the hormone control the digestions. So when the foods arrive the stomach, the hormone gastrin is a hormone released from the gastrin walls and released by the gastrin walls. And when the foods reach the stomach, they reach the gastrin juice. The gastrin will be released from the gastrin wall, and the gastrin is a hormone. Hormone means they need using the bloodstream, using the blood vessel to carry the hormone outside and go back up. So by the circulation back to the stomach to stimulate production of gastrin juice. So the food reach the stomach, the gastrin will be released from the gastrin wall and back through the circulation, go back to the stomach. Okay, so this positive stimulation. So by circulation back to the stomach to stimulate production of the gastrin juice. So the hormone in the control of the digestion So when the acid cheme reach the duodenum, when acid cheme arrive the duodenum, the hormone CCK is a whole cytokine and the secreted by the duodenum. So their hormone here, the CCK and the secretin released by the duodenum. The CCK, what's the CCK? CCK can stimulate the pancreatic digestive enzyme and the bile from the gallbladder. So when the acid cheme reach the duodenum, the acid cheme will stimulate the CCK. CCK will stimulate the gallbladder contraction to release the bile acid, the bile salt. And also the CCK can stimulate the pancreas to release the digestive enzyme. And the secretin can stimulate the bicarbonate to the neutral. Secretin can stimulate the bicarbonate to neutralize the acid cheme. So this is the hormone control the digestion. So the CCK released from the duodenum. And also there is another, when the cheme is rich in fat, if you intake a person intake the food and containing the large fat, rich in fat, the higher CCK and the secretin by duodenum will be produced. So the higher fat will produce the higher the CCK and the secretin. And the higher CCK both act on the stomach to inhibit the higher fat CCK can inhibit the peristalsis and the secretion of the gastric juice and the slowing the digestion. So this is the inhibit. So if the acid cheme contain the high fat, they will secrete large amount of the secretin and the CCK and inhibit the stomach peristalsis. So slowing the digestion. So the homeostatic regulation of the cellular fuels. So we intake that after the digestion, the glucose will be absorbed. So there is a hormone control the glucose homeostasis. This is well known. So the normal, I just very quickly to show the slide. So the glucose after we eat the foods, and the glucose will be dramatically increased, the blood glucose. So the normally blood glucose around the 70 to 110 milligram. So after the intake, the glucose level will be increased. So the glucose after the eating, such as after the eating, the glucose will be increased. So the pancreas will release the insulin. Insulin induce the smooth muscle cell or fatty cell, or induce the liver to storage the glucose, converted the glucose to the glycogen, and the storage the glucose as the glycogen. So the glucose the level will reduce to the normal level. In the fasting, after maybe a person already hungry for a long time, the blood glucose level decrease. So the pancreas will release the glucagon. Glucagon will catalyze the convert of the glycogen to the glucose and make the blood glucose level increase to the 70 to 100. So this is control the glucose is for the homeostatic regulation of the cellular fuel. So the regulation, the appetite consumption. So when we needed to eat, and when we needed to eat, and are you really hungry, or is it's already enough for you? So how to control? So in the hypothalamus, in the brain, there is a center called the satiety center. So is it possible to intake the more too much foods? So called the overnourishment. So overnourishment means consuming more calories more than we need, leading to obesity. And obesity contribute to the several human disease like the cancer, DM, diabetes, and the heart attack and the stroke. So appetite regulating enzyme is very important to control our intake, control our body weight, even control the disease happen. So there are several enzymes here. Either ghrelin. Ghrelin is secreted by the stomach work and triggered by the hunger and the lost weight, and the ghrelin level increase. So the ghrelin can enhance, stimulate satiety center, and tell the people intake the more food. So when people in the hunger or lost weights, and the ghrelin level will be increased, and to stimulate the satiety center to intake the more food. And the insulin secreted by the pancreas. After the meal, the insulin level increase. So after the meal, the insulin level increase. The insulin also can inhibit, tell the satiety center, the energy, the glucose level is enough. So don't intake the more foods. So the insulin can inhibit the satiety center. And the leptin is like the molecule secreted by the adipose tissues, also can suppress appetite. Another is the PYY, is a peptide secreted by the small intestine after the meal. The appetite suppressant counter the ghrelin. So this figure tells us the ghrelin can stimulate the satiety center and tell the people intake more foods. And the insulin, PYY, and the leptin inhibit the satiety center. Okay, so this is the regulation of the appetite and the food the consumption. And there is an animal. This animal is very, very cute, I can say, and already extensively used in the research, called the OB and the DB mice. So the genome OB, the mice with the mutation in the OB and the DB gene grow much more mass and than the wild type of mice. So you can see the OB and the DB positive, the average change in body mass about the 8.3, and the mutant, OB mutant, the OB mutant, they will produce make the mice bigger and the face and the grow faster. So the OB gene is very critical for the obesity or for the obesity related disease. Okay. And then so this is for many research study the obesity and study the obesity related disease like the hypertension or like the atherosclerosis, or using these mice to study. So we intake the foods and make a little bit complicated conversion of the foods to small molecule to intake. So finally we need to convert the foods to the energy, power the body. So the chemical energy powers the body. So the first one is ATP. ATP as the energy by oxidizing the small molecule. So the energy in foods is counted in the calories. So there is a term called the BMR, basal metabolic rates. Basal metabolic rates is the number of the calorie resting animal. Resting animal required to full essential process in a given time. So how to count the BNR? BNR, we cannot count the ATP because in the living animal. So in the research, the biologists always count the O2 consumption, because an animal using 1 liter of the oxygen needed 4.83 kilocalorie from the foods. So we can count how many liter of the O2 consumed in the animal, and to count the BNR. So for example, a body used 16 liter of the oxygen in an hour, and the metabolic rate is 16 times the 4.83, and the total number is 77.28 kilocalorie per hour. So I just want to mention we intake the food and digest the food using the enzyme and take a long time. Finally, we need the energy to power our body. So body fat and the fat the diet, the healthy women about 20% to 25% of their body weight in fat, and the 15 to 90% in the healthy men. So the women have the higher fat in the body, and the fat, those we cannot make essential fatty acid and get from the diet. The fatty acid, like the amino acid, we can divide the amino acid to the essential amino acid and the non-essential amino acid. The fatty acid, the same, essential fatty acid and the non-essential fatty acid. And the non-essential means we can produce by ourselves. And the essential means that our body cannot produce it, and we need to get it from outside. So there is a term called the RDA. RDA is a recommended dietary allowance. It's a mineral standard established by the nutritionist for preventing the nutrient deficiency. So there is a table here that you can look. So the healthy effect of potential problem, the extremely low carbohydrate diet. If the okay.
Yes, Prof.
Okay.
Yes.
Yeah. Okay. Maybe
Yes, Prof.
We can stop here. Okay.
Okay. So we already have question here, Prof.
Okay.
One student has already asked question about the vomit on the chat room, Prof.
Okay, no problem.
Yes. Well, actually, for dietary needs and all about nutrition, we also have another elective course, Prof. We call that as nutrition and health. So we also covers the discussion on RDA, and then also basal metabolism rate, and then needs for nutrition along a human lifespan, like that. So yeah, maybe for the discussion on digestive system until how the feces is excreted, maybe like that, yeah, Prof. Yeah, and then the regulation, usually we will talk in nutrition and health, like that, Prof. Okay, so there is one question already, Prof, from Dia. She would like to ask about vomit mechanism. How can a human vomit the food? What is the mechanism of vomiting? Does the mammalia has the digestive system also, which has a sphincter that keeps the food from returning back, so prevent to have vomiting rather than human usually do, like that?
You mean the foods in the mammalia or something to relate it to the health or some?
To vomit, related to vomiting, Prof. Vomiting.
Vomiting.
Yes. Does mammalia has a sphincter just like in human?
Yeah.
That will keeps the foods and then prevent it to return back, and then no vomit will occur, maybe like that in mammalian.
Yeah, the vomiting. Yeah, the vomiting is it's always occur in the herbivores, I think. Herbivores animal need the vomiting because they intake the plants or grass or something. They have a cell walls, so needed to vomiting to digest the cellulose. But in the vomiting, is it the vomiting like the cellulose digestion needed, right?
Yes.
Yeah, and I'm not sure, but in the human, in the mammalia and in the human, we needed to fermenting. Yeah, I think many products are fermented, right? Like the milk or something. But it's healthy. But how can keep the bacteria living in our body and cannot be killed or something? It's a good question, but I needed to find some evidence to say that.
Yes.
Yeah.
Okay, we'll be okay, Prof. So I think we also have some of our colleagues from human biology and physiology as well. Ibu Yulilina maybe would like to ask some question. Ibu Nurmasari and Bu Ratna, Bu Ola, Bu Ninul, Bu Ratna. Any question to Dr. Chen?
Yeah, I think I speak a little bit first. So maybe,
Yes.
yeah, if you have any question, you can send me the email or something. Also, you can ask you if my email is not so smooth, not workable, maybe send to the Jack. Jack, I will reply via the email. Is okay. Yeah, if you after the work after the course, maybe all the professor and the student or faculty has in, also can send me the email. I can reply.
Okay, thank you very much, Dr. Chen. Maybe this is very interesting about the OB and DB genes. This is only mutation only happen in mice, Prof?
Yeah, happen in mice. In human, maybe it's using the genetic manipulation. So to make the OBE mutation. So this is an animal model for study the obesity and the obesity related to human disease.
Okay, this is only the model of obesity. Yes. Okay. Any other question from class? Ada yang mau tanya lagi? No? Okay, from Haris. Haris, ayo, come on. You can ask Dr. Chen. Haris. Yes, Haris?
Yeah.
Okay, please.
Okay.
Professor, I want to ask, how is the carnivore separate the meat and the bone due to they eat the prey in the big chunk?
Excuse me, what the carnivores?
Carnivores, Prof. Carnivores.
Carnivores?
Carnivores, the animal who eat meat.
Yeah.
How will they separate meal from the bone? Is that like that?
It's the carnivores. Yeah, carnivores eat the meat.
Yes, ma'am.
The meat.
Can you specify the question, Haris?
The carnivores eat the meat. Maybe they use the meat as the food sources. They don't eat the grass or plants. Yeah.
What is that? The question, Haris?
I want to ask if how they separate the meat and the bone, because they eat a big chunk, like maybe the bone is still in the between the meat, so they might swallow it into their.
Oh, separate the bone and the meat. Yeah, I know.
Yes, Professor. Yes.
So how to separate the bone and the meat? I think it depends on the different animals. Some animals can eat the bone. I think like the dog. I'm not sure. So the bone and the meat, how to separate them in the carnivores? They always use the different. I have mentioned the different teeth structure. You know, the carnivores has a special teeth structure here. Here, this one. So the carnivores, the teeth structure is different. They have the very big the canine, so they can spread the meats. Yeah, you mean how to separate the meat? How to separate the meats and the bone?
So this is their fit action maybe, Prof.
Fit actions.
No, the teeth will help to separate the meat from the bone, from what they are eating. Is it like that? Because
separate
of
what they are eating, the meat, how to separate the meat and the bone. Yeah, maybe the carnivores eat using the tooth or using the canine.
Yes.
To try to separate the meat and the bone. Because the molar and premolar and the molar not so important for the carnivores. The carnivores always use the canine and the incisor to try to spread to remove to take out the meat. Yeah.
Yes. That's why it is a very large, the incisors, yeah, Prof.
Incisor and yeah.
Mhm.
Okay.
Yeah, maybe like that, Haris. Karena ini incisors-nya cukup besar. Jadi itu the action will be taking part by the incisors, maybe, I think.
Yeah.
Yeah. Okay.
Thank you, Prof.
Thank you. Yeah.
Okay, any other question, class? Any other? No, if no more question, or if you feel shy to ask question, you can ask to
Okay, no problem.
you can
Yeah.
write me the question through our class coordinator, then I will pass to Dr. Chen through
Okay,
Dr. Chen's
welcome.
email, if it is okay, Dr. Chen.
Yeah, I think in addition to the digestion, maybe if all the faculty or the professors, students has a question related to the research or to other parts, we can discuss. I think the research or teaching is mutual. I mean by directional. So
Yes.
yeah. So we can discuss and anything is okay.
Okay, so maybe I would like to ask for your permission. Maybe I will share your email address to the faculty here, Prof.
Yeah, no problem.
So maybe,
no problem.
yes,
Yeah.
so maybe we can share what is that, collaboration on research. But if I see that you also concerned about tumor and cancer and the treatment with natural product, yeah.
Yeah,
Prof.
this is my research topic. Yeah.
Yes, we do a lot of works on that field actually in our faculty. So maybe we will have to discuss more about the collaboration.
Oh yeah. I'm great to do that. Yeah. So yeah, I visited Indonesia maybe several times, maybe two years ago. So so I think we can discuss further. Also, if you have some special, very interesting topic, we can discuss. I also can find the faculty in the TMU can make the connection.
Yes, of course, Prof. We hope will be like that.
Yeah.
Okay.
Thank you.
Thank you very much,
Thank you.
Dr. Chen, for today lecture. Hopefully, we will meet again next week.
Okay.
Next Tuesday. With urinary system, Prof.
Yeah.
Okay. Maybe, Prof, you would like to say something before we end the session today?
Yeah, it's okay. I already say. It's okay. Thank you.
Okay.
Jack, is Jack online?
I'm not sure. I don't think so, Prof.
Okay, it's okay. Yeah, thank you. Maybe we can close here. Yeah.
Okay, so we will close here and we will meet again next Tuesday at 10 p.m. Jakarta time to discuss about urinary system. Okay.
Yeah. Okay. Thank you.
Thank you very much for your time, Prof. Good afternoon.
Good afternoon, everybody.
Okay. Bye-bye, class. Good day.